Okay, thank you very much. Hopefully this will work here. We had a little bit of an issue with my using Keynote instead of PowerPoint and other tools, but we'll see if this works. And yes, my title is insanely long. I think I answered their email about my title at about 2 in the morning one night. So what I want to talk about is a little bit about my research on the evolution or evolutionary studies of microbiomes, but also talk about the concepts of microbiomes, or not really the concepts, but how we communicate about microbiomes. And that's where I do think it is actually true that microbiomes are frequently ridiculously overhyped and frequently horrifically underappreciated. And that's where I came up with the title. So bear with me. I've never done a talk where I've tried to combine these two things together into one talk, but we'll see how it goes. So as I assume everybody here knows, but I'll just reiterate, the microbiome has, over the last 30 or so years, become an incredibly hot topic. It's not that people weren't studying communities of microbes previously, but it's become more commonly studied to study entire communities of microbes that are associated with some type of host in particular, but also free-living communities of microbes. And this has, become a very, very hot topic in the news, in the scientific literature. These are slides from a few years ago. I didn't remake them, but I went and looked. It's still sort of an exponential, or not quite exponential, increase in PubMed hits to papers with microbiome somewhere in the abstract or the title. And what I thought I would take a step back for a minute, and many people listening probably have some appreciation of this, but I just thought I'd go through what I think are some of the reasons that the area of microbiome studies has become so popular, so pervasive. And one of them I think is really important is that there's been a growing appreciation over the last 30 years, certainly, maybe 50 years, but accelerating appreciation of the importance of the diversity of microbes, the functional diversity, the evolutionary diversity, the ecological diversity of microbes. And that's not just the ones that make us sick, which have been studied for quite a while, as we're all dealing with right now, of course, microbes that make us sick, but also the microbes that provide benefits to various organisms, whether it's nitrogen fixation in legumes or energy acquisition in various animals, or things that we use microbes for making our food, or microbes playing important roles in global nutrient cycles and in the health and well-being of the planet. There's just been a growing appreciation of the importance and diversity of microbes over time. And I think that this is one of the contributing factors to the rise of the study of microbiomes. Second, which people may not appreciate, but I really think about this a lot. So prior to working on microbiomes, I worked at a place called Tiger, the Institute for Genomic Research, where we were sequencing genomes back when it was expensive and somewhat difficult to sequence a genome. And I became concerned at the time with people who were overselling the benefits of genomics, in particular the human genome, but also other genomes of various organisms, where people would say, the genome, all we need is the genome sequence of this organism, like humans, and we're going to suddenly revolutionize, cancer treatment or agricultural studies, or, pathogen prevention, pathogen killing, and other things. And a lot of this was just overhyped. And when the human genome was done, and when other genomes were done, people suddenly said, oh, now we can move on to these other things that are actually the important things. These are the ones that are going to revolutionize everything. And it was the transcriptome or the epigenome, or some people refer to the variome, that is polymorphisms within populations. And the microbiome was yet another thing. If we can't explain all of human Drosophila, Arabidopsis, corn, whatever biology from their genome, maybe one of these other things explains that biology. And the microbiome was one of the things that was a candidate for explaining a lot of the biology of organisms that you couldn't glean just from looking, staring at, and dealing with the genome sequence. So I call this the post-genome blues. time period. Clearly, another major component of the rise of genomics and advances in life sciences, but in particular in studies of the microbiome, have been technological advances that have allowed people to study entire communities of microbes and study many samples of those entire communities of microbes and start to try to get an idea as to what functions are present in those communities of microbes. I mean, an entire community of microbes has a lot of participants in it, regardless of where you're looking. In the human gut, it could be thousands of species, each that come in many different forms, and there's three-dimensional structure to this, and there's all sorts of different proteins being produced and metabolites being produced and functions being produced, and it's just very, very complicated to study this, and therefore technology has helped with this. The technology that I have spent a lot of time taking advantage of, but it's not by any means the only technology that's important here, has been DNA sequencing. Clearly, the improvements in speed, the decrease in cost, the increase in throughput of DNA sequencing, and our ability to analyze DNA sequence data or RNA sequence data has revolutionized many areas of biology, including microbiome studies. Another really important thing is the growing appreciation that the microbiome, the community of microbes, might be providing important functions to organisms beyond that of the single microbes that we had been studying for many years. This was sort of captured in this classic figure from a study from Peter Turnbaugh and colleagues in Jeff Gordon's lab of the obese mouse, where you could transplant the microbiome from this mouse that was genetically had defects that led it to become obese. And when you transfer the microbiome from those mice to germ-free microbes that don't have germ-free mice that don't have the genetic defect, at least in this original study, those mice got some amount of obesity, and that therefore the microbiome was transferring the function. This picture, this image, was shown at dozens of microbiome planning meetings, at meetings where the NIH was planning the big human microbiome project. It was probably shown in various other places. I went to the NIH-funded ones, and it was, people were very, very excited, not just about this, but of dozens of other studies that had shown that the community of microbes might be important for allergy and autoimmune diseases and behavior and obesity, and in animals, and then in plants, in yield and in nitrogen acquisition, and so on. And so people got very excited about the functions of the microbiome. That is, the microbiome, could do something that was interesting and important. It wasn't just a collection of microbes hanging out and doing nothing. And this wasn't just humans. An important thing was that people were showing this in lots of other animals, in plants, in a variety of other systems. And there were even studies expanding into the community of microbes that were in buildings, or the community of microbes that were in various other places. There's this great excitement about communities of microbes generally, and the functions that they might provide to organisms or to the planet in some way. And this is where I come in. In my lab, we've been studying microbiomes for about 20 years now. I'm very excited about microbiomes, although, as we will see at the end of the talk today, I'm also think we need to have some serious skepticism about what's going on in the microbiome area. And I just thought I would walk through, I've lost my animation here. Hang on one second. Well, I guess we're gonna have to deal with possibly no animation in my slides. Hang on one second. I'm just gonna see if I can turn back on the animation. Well, maybe I won't have animation. The perils of switching. Do you not see my slides anymore? I don't know if anyone can answer, but
no, I don't.
You don't?
It's a block. Oh yeah, it's coming. Your slide.
Yeah, I'm gonna have to disconnect my other monitor and just hit play. If you just hold on 1 second, because the keynote thing did not work here. Now, can you see my slides?
Yes.
It's just phylogenomics and evolvability.
Yes. But it's
Okay.
yeah.
I'm gonna have to do it this way. Sorry about that. Back to the talk. So what I work on, what I'm interested in, is not specifically about microbes or microbial communities. What I'm interested in is the origin of new functions in novelty. And there's a simple way that I represent this. If you have an evolutionary tree showing a group of organisms, imagine you have an ancestral characteristic, the organism is red or something like that. One of the descendant lineages here keeps that trait. Another descendant lineage keeps that trait. Another descendant lineage keeps that trait. But there's a change in the function of this organism where there's a novelty originates on this branch. And what I've been interested in for many years prior to working on microbiomes was how do those novelties come about by changes inside the genome of an organism through mutation processes, duplication, deletion, rearrangement, recombination, and so on. And in particular, I was interested in why these processes, all of those processes that led to the origin of novelty, differed between taxa, something I call evolvability. What leads to this variation in these processes? And I invented this term, phylogenomics, which I use for my Twitter handle and my blog. And I'm sorry I invented an omics word because now I kind of hate new omics words. What I'm interested in is integrating genome data and evolutionary studies to help understand evolvability. And again, I work on this for many years with intrinsic evolvability. But over time, I slowly shifted to being more interested in how organisms acquired novelty from the outside, something I call extrinsic novelty or extrinsic evolvability. So one way that they can do this is through lateral gene transfer. An organism can take the genes from one organism and use it through recombination or gene transfer, acquire the traits that that organism has. And what I've been interested in for the last 15 or 20 years has been, forget about how you acquire DNA from other organisms. How do you acquire functions by interacting with other organisms through some type of symbiosis, either with one organism or multiple organisms or communities of organisms, microbiomes, that is. So that's what I've been working on. That's the evolution part of this talk, is what I've been interested in is the evolution of microbiomes and how we can use evolutionary studies to better understand these communities of microbes and how they enable organisms to acquire functions, not by stealing genes, not by changing their own genome, but through interactions with other organisms. And so I sort of organized this under the concept of sort of one big topic area, microbial phylogenomics and evolvability. And I have major research projects in this area. We spend a lot of time developing what I call phylogenomic methods and tools and resources and reference data. And we also are very concerned with communication and participation in microbiology. And so what I'm going to do right now for the next few minutes is take you on a tour of a little bit of the work we do before talking about the last topic, which is going to be communication about microbiomes. So I'm going to give you a really high-level tour of research projects and then a little bit more detail on some of the phylogenomic methods and tools. We don't study any one system in my lab. I'm interested in the rules by which communities of microbes interact with each other, and I'm very interested in studying different systems rather than one system. So I'm going to give you this brief high-level tour of what we work on, in particular with the recent work on extrinsic novelty through symbioses. So most of what we work on can fit into this, what's called the host microbe, what I call the host microbe stress triangle, where there's a host that's interacting with a microbe and then some stressor that affects how the host and microbe interact with each other, or how the host and the microbiome interact with each other. And what I'm interested in is when you put organisms under some type of stress where novelty would be beneficial, what leads them to evolve certain types of novelty? What leads them to change their interactions where symbiosis or some type of interaction can be a solution? And then, more importantly, how can we manipulate this and or to force new functions upon systems when we need to? So one of the types of systems that we work on is where the stress is some type of nutrient stress. Lots of people work on nutrient stress in hosts and how microbes or microbiomes can help compensate for those nutrient stresses. I'm just going to give you a few examples of some of the systems I've worked on. Actually, when I was an undergraduate, I worked with Colleen Cavanaugh on chemosynthetic symbioses, where There's some type of marine invertebrate. They have endosymbionts that live inside of them, and those endosymbionts help those organisms acquire carbon from the environment, or sometimes other nutrients from the environment. And this is how I got interested in microbes originally, and how I got interested in evolutionary studies, because as I'll talk about in a little bit, we used the studies of the evolution of ribosomal RNA genes in order to figure out what types of organisms were living inside these chemosynthetic systems. You could say I graduated later on to working on a system that was also a nutrient requiring system in a collaboration with Nancy Moran's lab, where you have insects that are obligate sap feeding insects. Many people are probably familiar with the aphids, where they have symbionts that make amino acids that they can't get from their phloem sap that they feed on. Nancy Moran became really interested in the insects that obligately feed on the other circulatory system of plants, the xylem, which is more nutrient poor. And we collaborated with her working on this system where there were 2 symbionts that lived inside this insect, the glassy-winged sharpshooter. And we used genome sequencing and evolutionary studies to try and understand those 2 symbionts and how they help with the nutritional needs of this insect. We've done some work on nitrogen fixation in communities of microbes. There's, for example, a variety of corn that grows in the mountains of Mexico that has been of interest to many groups that may be sort of evolving or have a symbiosis with free-living nitrogen-fixing microbes that help supplement the nitrogen in its diet, not in the way that legumes do it, but maybe as a simple low amount of supplement to the nitrogen in its ecosystem. Another one that we've been really interested in, this is sort of generous to call this a nutrient study, but it sort of is how animals produce odors and how some animals may manipulate that in order to affect behavior. We've been working with Holly Gans, originally started this in my lab with David Coyle and a few other people on the anal secretion glands in cats. And it looks like similar to what other people have shown in other animals like hyenas. There's been a lot of work by others in hyenas and other animals. Looks like cats are secreting some type of what you could say is food for microbial communities, and then the microbes grow in that food and produce odors, and that leads to each individual cat having a distinct smell because they each have different microbes. And what we've been working on is trying to figure out what are the microbes that are living in any of these systems, what volatile compounds are they producing, and eventually we're really interested in how that might affect the evolution of behavior in the Felidae. I have a new postdoc, Connie Rojas, who's working on a project related to this. So this is an ongoing project in the lab. Another host microbe stress triangle that we, and of course many other people, spend a lot of time thinking about is where you have a host, a microbiome, and some type of pathogen. And the question here is, does the microbiome in some way affect the pathogenicity, the virulence, the survivability, and other features related to the interaction between the host and the pathogen? And we've done this in a lot of different systems in my lab. We've worked in collaboration with multiple people on studying flu, duck, gut microbiome interactions. We've had a grad student, Katie Delhausen, who was really interested in koalas and how koalas, which have an obligate symbiosis with microbes in their gut that's still poorly understood, that helps them detoxify eucalyptus leaves. There's also a chlamydia epidemic going around in koalas, and those koalas are treated with antibiotics, and then many of them get sick, probably because their gut microbiome is decimated by the antibiotics to treat the chlamydia. So she was interested in trying to figure out what's happening in these koalas that are treated with antibiotics, and whether or not we could understand what gut microbes might be affected by that antibiotic treatment in order to then eventually restore that in other koalas. I have 2 current graduate students, Sonia Ghosh and Marina De Leon, who are working on this triangle that a lot of other people are very interested in between frogs and the chytrid fungus BD that has been decimating amphibian populations throughout the world, and whether or not some of the microbes in the skin can provide some protection to those frogs. A third type of this host microbiome stress triangle that I'm interested in is when the environment is changing in some way, whether that's through domestication, for example. We've done some work on the rice microbiome, or one project that we're still working on now is where the change in environment in this case was plants moving from the freshwater back into the marine environment, into the oceans. These are the seagrasses. They're the only plants that live fully submerged in the marine environment. There have been 3 separate invasions of the marine environment by freshwater plants, and we're interested in what's happened to the microbiome in these situations and whether or not that provides any insight into either terrestrial plant microbe interactions or the biology of seagrass. And again, we have a current project going on right now between my lab and Maggie Sogan's lab and Jay Stackowitz's lab, and a postdoc working on this, Gina Chaput. Another project that's just wrapping up is a collaboration with the Smithsonian Tropical Research Institute, where in this case, the change has been the rise of the Panamanian Isthmus. And the question has been, what has happened to the microbes and the microbiomes of all the organisms that were separated and then led to new speciation events on the 2 sides of the Panamanian Isthmus? And can we use these thousands and thousands and thousands of speciation events to better understand how microbiomes respond and evolve over periods of time? Okay, so now switching to from the tour of research projects to the tour of phylogenomic methods and tools. And again, it's sort of a brief tour. I just want to give you the perspective that a lot of what we think about is not how do microbes evolve, but how can we any evolution as a tool to better understand the biology of single microbes or communities of microbes. So many people are probably familiar with a lot of the work in this area that other people have done. We're not by any means the leaders in this, but we've done some work on using ribosomal RNA and in particular phylogenetic studies of ribosomal RNA to better understand communities of microbes. This again started when I was an undergraduate, trying to understand this one symbiont by building evolutionary trees of it. And a key point here that I hope everybody remembers is that similarity is not the same thing as relatedness. You can't just do a blast search with something, get a % similarity and figure out what it's related to. You really need to build an evolutionary tree. And so we've spent a lot of time trying to design tools that allow you to automate this. Danying Wu in my lab built a tool called STAT many years ago. Amber Hardman in my lab built a tool called Waters, all with the same purpose to automate building of phylogenetic trees. And there's lots of other things that we've been in collaboration with people to do to try and add in evolutionary analysis into studies of ribosomal RNA, whether that's for finding OTUs or for dealing with the copy number variation that occurs between ribosomal RNAs, where you can actually correct for some of this using phylogenetic trees. But probably more importantly is we've done a lot of work on trying to use other genes as markers for ecological and evolutionary studies of communities of microbes. And so this started with some of the early shotgun sequencing that people did. I collaborated with Craig Venter and others on the Sargasso Sea and then the GOS studies and designed tools to, for the, what for us was the first time where you could get sequences of protein coding genes from random samples of environments and compare them to ribosomal RNA and the phylogeny of those. And in many cases, the data that you get from those is actually better than what you get from ribosomal RNA for a variety of reasons I'd be happy to talk to people about. So we've done this with first 5 marker genes and then 30 marker genes with a tool called Amphora, and then a few more marker genes as we got more and more data. Aaron Darling and other people in my lab built a tool called Phylosif that's highly automated this. You can use this to then do phylogenetic diversity studies of communities just like you would do with ribosomal RNA. Another thing we've been very interested in is how do you predict functions of genes using evolutionary tools? And if you want to understand how functions evolve, you need to be able to predict functions well from sequence data because most of the data we have is sequence data. And again, key thing here is similarity does not equal relatedness. So if you want to predict the function of a gene, it's better to build an evolutionary tree of that gene than to do a blast search. That's sort of the simple lesson that has come from this. This is the same principle that applies to ribosomal RNA, but now applied to protein function. Tom Sharpton and Katie Pollard developed tools to automate this. Others have also done this. Another thing that's really important and a useful tool from an evolutionary point of view is something called phylogenetic profiling, where you look at the distribution patterns of genes across organisms and use that to try and predict interactions between genes rather than interactions between organisms. We've done this in particular with sporulation genes, working with Rich Lozick and other people in Rich's lab to predict the which genes are involved in sporulation across the gram-positive sporulating organisms. And then the last thing in terms of phylogenomics and evolvability is, if you want to study organisms, you need to understand their evolutionary history. You need to know what they're related to. And it's really better to do this rather than with single genes, to do it with the whole genome if you have the whole genome available. And so a lot of what we've done is try to build tools for inferring phylogenetic trees of organisms from concatenated alignments of protein marker genes, where you've shown that these are good, useful single genes, but then you concatenate them together with these various tools. I'll just skip over that. What I'm not going to talk about is another thing that's really important, is linking phylogeny and function. There's lots of things that you can do there where evolutionary analysis can improve your ability to bin metagenomes, for example, from an environment, which helps you link phylogeny and function. In terms of resources and reference data, it's sort of the same approach. Evolutionary studies can help guide the generation of reference data, which in turn can then improve your ability to study microbiomes. The most obvious way that we've done this has been to try and generate genomes from organisms from novel branches in the tree of life compared to what genomes are available. I started this previously when I was at Tiger with an NSF Tree of Life project. I helped lead the Genomic Encyclopedia of Bacteria and Archaea, the GIVA project that was done at JGI and is still has many spin-off projects still going on at JGI, where we basically marched our way through the bacterial and archaeal portions of the tree in collaboration with the German culture collection, the GSMZ, found cultured organisms for which there were no genomes available on their branch, and then sequenced those genomes and shared them to the community. And we've done this for individual clades like the haloarchaea, cyanobacteria, and then most of evolutionary diversity of microbes is not present in cultured organisms, so you need to go after uncultured organisms in order to fill in the tree of life. And we're not, of course, the only ones doing this. This is where MAGs, metagenome assembled genomes, come in. Lots and lots and lots and lots of people are filling in the tree of life by going to environments where you don't have to culture the organisms. And if you can get a good genome assembly or a MAG out of an environmental sample, you can now help fill in the reference genomes from across the tree of life. The same is true for protein families. It's also important to build up resources for the diversity of protein families across the tree of life. And in some cases, some of those protein families can be used as good evolutionary markers. Not all of them can be used as good markers. All right, so that's the biology part. But what I really want to end with talking about is this communication issue that I raised before. So I think all of the studies that we've done on microbiomes, the key lesson that has come out of this is that community microbiome host interactions are just way more complicated than single host microbe interactions. They are much harder to study. They're much harder to manipulate. They're much harder to make inferences about. They're much harder to understand the evolution and ecology of these interactions. I mean, when you have 10,000 taxa, each coming in hundreds of different strains, each with, living in a slightly different niche, say inside the gut, and then in humans, there are different microbes that live on the skin and the mouth and the vagina and every other part of the organism. They differ over time. They differ between individuals. I mean, it's just so complex, so incredibly complex. What I want to talk about for a few minutes here is this issue of communication about microbiomes. So if we go back to this, yes, it's been very, very exciting. Lots of people working on microbiomes. Lots of amazing work has been done on microbiomes, and I think they're very important to the biology of the planet, to the biology of every plant and animal and other multicellular organism, as well as single-celled organisms on the planet. But there's a big downside, or there's actually two big downsides. And this is what I used to call microbiomania versus germophobia. So with germophobia, we have this huge problem, which is that everybody wants to kill all the microbes or is scared of all the microbes. And with what I call microbiomania, we have this opposite problem, which is that some people think that all microbes are good and that we should embrace all microbes across the planet because they're all beneficial and they're all going to help us do everything we've ever wanted to do with microbes, with life on the planet. And of course, neither of these are really true. So the germophobia side of things basically says all microbes are bad. We should put antimicrobials in everything. You should avoid all microbes. You should avoid animals too, like pets, because they have microbes in them. And there's something called Swab stories that I'll come back to, that you should avoid germs anywhere. And with microbiomania, the opposite is what people are saying. All microbes are good. You should use probiotics in everything. You should embrace all microbes. You should lick your animals, or even lick the poles on your subway system, because you should embed every microbe on the planet into your system, and fecal transplants will save the world. And this is basically the underselling and overselling that I used in the title of my talk, that I think there's many people, the press, snake oil salesmen and others, and scientists overselling the microbiome, and there are many that are underselling the microbiome. And we need to meet somewhere in between. So I just thought I'd walk through a couple of examples of this, of how overselling happens, or the problems with overselling. One of the first ones, and ones that lots of other people have pointed out, is that correlation does not equal causation. Some microbiome correlations with health states are actually due to the microbiomes playing a causal role in that health state. That's the excitement of microbiomes, but most are not. Most are not due to causal connections. And I don't know if people saw this recent study suggesting that the autism spectrum disorder microbiome association is not a causal connection, and that it is that autism affects the microbiome, not the other way around. That just came out about a month ago. Another issue with overselling the microbiome is issues of contamination. There have been many studies that have shown that reagent and laboratory contamination is a big issue that we need to worry about, and not enough people worry about it. Some observations of microbes being present in a system are mistakes. There's been many suggestions this is the case for the claimed placental microbiome, and it should be viewed as a cautionary tale. There's another part of overselling the microbiome that's really important, that really was brought to the front by a paper by Toby Hammer and others, that basically said that caterpillars lack a persistent resident gut microbiome. They have microbes there, they're present, but they're just not important, was the conclusion from that study. And we need to realize that just because microbes are present somewhere, that doesn't mean that they're doing anything interesting or important in that system. Another aspect of overselling that's really important is that not all non-pathogenic microbes are good. So for example, I took these pictures a few years ago when I went into CVS drugstore one day and saw this display of probiotics where they had, microbes that for which most of them had no scientific studies behind them. And then they had a section, here are the probiotics that you should take if you're an adult. Here's the group that you should take if you're a kid, a senior. Women should take different probiotics. I mean, it was like basically just spewing out bacteria are good and we're going to sell them to you. It's not that probiotics aren't useful. There are some studies that show that some probiotics work, but you can't just throw a non-pathogenic microbe at something and say, hey, it's going to be a probiotic. And this may seem trivial. These probiotics they're selling probably have no effect, but there are many people that have gone way further than this. For example, there were two people at my institution many years ago at UC Davis who claimed that they were using a probiotic therapy for glioblastoma, where they injected some bacteria into people's brains because they thought they would have a probiotic effect, and they killed them. So probiotics are mostly oversold, unfortunately, even though some are useful. Another thing that's really important to realize is that just because we can study the microbiome in people, just because people differ from each other or differ over time, does not mean that there's an era of personalized microbiome medicine coming anytime soon. Again, there are some studies that say that we can look at someone's microbiome and say something about a health condition that they have. But most of the claims in this area are bogus. If you're familiar with uBiome, you might want to listen to the podcast made by the Wall Street Journal about uBiome. For full disclosure, I was on the scientific advisory board of uBiome. I quit before many of the problems at uBiome, but I confess I was swindled by some of the overhype of the potential of just diagnosing a microbiome and how it might be useful to people. And most of the claims that companies make related to diagnostics and microbiomes are not backed by science. Again, there are some that are, but most of them are not. And then a last thing about overselling is that some microbes are bad. It's important to realize that some microbes are bad. So there's been this thing, the hygiene hypothesis, that basically says that, you know, we're missing a lot of microbial interactions, and that might have led to many of the modern problems of humans and society with obesity and allergies and autoimmune disorders. And that is almost certainly partly true, that some interactions with microbes have gone away, and that's been bad. But that doesn't mean that all microbes are good, and you need to go around the world eating dirt or licking the subway poles. Or, for example, there was a recent one from a UCSF doctor saying we should all get the flu because flu benefits your microbiome. So, you know, there's a litany of overselling issues. I'm not going to go through all the other ones. I'd be happy to talk to people about this if you want. But there's a litany of these overselling issues. And then on the underselling side, there's also a litany of issues. The first one is just kill everything. We should kill all microbes. And that's clearly underselling the benefits that come from microbes. If people are saying we should put antimicrobials in everything. There's antibiotics in many toothpastes. We know that antibiotics are being overused in human medicine and animal medicine and in various other places on the planet. There are antimicrobial coatings on food, on cars, on shoes, in buildings, on clothing. There's antimicrobials everywhere. And this is almost certainly, on average, really, really bad. Just trying to kill everything because there's this general idea that germs are bad. We've gone crazy with this. And there's another area of this that's very related, which is what we call swab stories, or actually Mark Martin is the one who introduced this to me, the idea of swab stories, where you go to some place, you swab and you grow the microbes there and you say, oh my God, there are bacteria all over the phone or the shoes or the doorknob or the TV screen or the wherever else and the fast food restaurant, and we're all going to die because there are microbes there and we should kill them all. We have to kill them all. We're all going to die. And it's just crazy. Like, we've gotten this approach where, you know, all microbes are bad. It absolutely undersells the potential benefits of some microbes. And this, you know, sort of germophobia has led to many, many, many bad things throughout society. And there are other underselling issues. You know, just because some organism is related to a pathogen does not mean it's pathogenic. Another big one is microbes with subtle effects have basically been ignored in most systems, and microbiomes have been ignored in many studies of plants and animals throughout biology. Just one that I wanted to add here is in conservation biology, microbes are generally ignored, but they're clearly very important for protecting the future of the planet. So there's this massive, you know, underselling and overselling of the microbiome, and I think we need to come somewhere in between to this. So what are the solutions to this? Well, one is to complain. I complain a lot. I give out an Overselling the Microbiome award, or I used to when I had time, on my blog. And I could really give one out every single day if I wanted to. There's overselling of the microbiome that happens constantly in the press, by scientists, by the public, by charlatans, by others. I complain a lot. So if you want to go look at microbiomania.net, and I have a list of all the awards that I've given out. It's not just me. There are lots and lots of, I've collected some of this on my blog, lots and lots of other people complaining too. There are many papers that people have published. Bill Hanage, Liz Bick has done this. Lots of other people have complained about overselling of the microbiome. Another solution is education and outreach. I think education related to microbes is really, really important for the public to reach out and try and engage people in communicating about microbes and the microbiome. I love this. This is David Coyle, who is my lab manager. He hates those swab stories, so he's written lots of articles, and he's like the go-to person to come to say that actually, yeah, when you swab a toilet, you find microbes, but it may not be as gross as you think. Another really important thing is citizen science. We can actually engage the public in doing studies of microbes and getting them to think about the good and the bad of microbes. And engaging students too is really important. It's not just the public, but trying to get high school students, undergraduates, and others thinking about microbial diversity in rigorous ways is really, really important. And I think we need to do more of it. So with this underselling and overselling of the microbes, what I'm hoping for is that we can end up somewhere in between where there's some balance. And I'm hoping that microbiome communications can evolve much like microbiomes may be evolving, but I'm hoping that they evolve to become balanced, even though, as far as I can tell, most microbiomes are not balanced. So I will end it there. Sorry about the slide complexity issue. I blame Keynote. And thank you all.